Good morning. It's me from BioWorld. In today's video, I'm going to start off with asking you to observe these two pictures. What is the one word you will use to describe what you see? Think about it for a bit. Yeah, I'll share with you what are the words that pop into my mind. Starting with the picture on my right here, I have words like dry leaves, falling leaves, brown leaves, dead leaves, or the season of autumn. Any one of your words there in the screen? Well, I hope so. Let's move on to the picture on my left. I don't have that many words to describe over here, but based on the leaves, I can see that the wind is blowing very strongly. But surprisingly, you see none of the leaves are falling. So I'll consider that the leaves are intact. That means they are attached to the plant or the tree. And the color of the leaf, of course, is green. Now, why would I want to introduce these two pictures to you today? That's because I'm now going to talk about a process in the plant known as senescence. Senescence means the plant is growing old. It is aging. Previously, in my videos on auxin, cytokinin, and gibberellin, we were talking about how the plant was growing. So that is the stage where the plant is still young. So today's video will be about the aging process in plants. The first hormone is abscisic acid. In short, they label it as ABA. This is its structure, which as usual, we don't have to memorize. And now we look at the effects of abscisic acid to the plant. Once again, I use this diagram from my Introduction to Plant Hormone video. And you can see, although it's blur, but you can see that the abscisic acid is required for two functions. That is abscission, that is where the name abscisic acid comes from. And the second function in this example here is seed dormancy. But we will be talking about dormancy in general. Let me start with abscission. Naturally, abscisic acid is going to promote abscission. This is antagonistic to the previous three hormones, auxin, cytokinin, and gibberellin, which were inhibiting abscission. These hormones prevented flower, leaves, or even fruits from falling off the stem. As in the picture with the green leaves you see, when the wind was blowing so strong, nothing fell. But abscisic acid promotes abscission where the leaves, flower, or fruit will fall off by itself. So if you saw in the picture I showed you, there was no wind blowing, yet the leaves were falling. So that is abscission. The second event is dormancy. Dormancy is when the plant sort of goes to sleep. The plant stops growing. To stop growing, the first thing is the cell division must stop. So abscisic acid actually inhibits cell division, and in doing so, it will suspend growth. Suspend means stop the plant from growing. This is necessary during seasons which are not suitable for plant growth. For example, during winter or during dry seasons. So dormancy can be useful to the plant too. This is antagonistic to the two previous hormones, that is auxin and cytokinin, because remember, auxin and cytokinin are necessary for cell division. Only with cell division can the plant start to grow. So abscisic acid works directly opposite to auxin and cytokinin. Besides making the plant dormant, abscisic acid also makes the seed dormant. Seed dormancy is when the seed does not germinate. This is antagonistic to gibberellin. Gibberellin promotes seed germination, that is for the seed to grow a shoot and a root, but gibberellin inhibits seed dormancy. Abscisic acid does the opposite, that is, it will promote seed dormancy, meaning that it will encourage the seed to not do anything, or in other words, it inhibits seed germination. I stress these words because in objective questions, students tend to focus on the word inhibit and promote without paying attention to the word dormancy or germination. So that is why I'm highlighting it here. Now, another thing related to dormancy is closure of stomata. Abscisic acid also helps protect the plant when under water stress, by helping the stomata close. So these are the roles of abscisic acid in plant development. Let me now explain the process of abscission in more detail. To understand abscission, we need to understand parts of the plant. This part here, where the stem of the leaf or stem of the flower or stem of the fruit is attached to the main stem, is called the abscission layer. Now the branch that attaches to the leaf, flower or fruit is called the petiole. So if we look at the arrangement of the cells around the abscission layer, it will look like this. You have your vascular tissues, and then you have all the cells here. These should be your parenchyma cells. And you see at the abscission layer here, there are very small cells that are closely packed. Now, when auxin, cytokinin, or gibberellin are present, they promote the cell division of these abscission layer cells to always be present. But when abscisic acid concentrations increase, these cells stop dividing and slowly they begin to shrink. Once they shrink, the abscission layer here becomes loose. And this will enable this part, the petiole that is connected to the leaf, flower or fruit, to fall off easily. Let me share a video to show you. In this video, I want to show you how easily it is for a leaf, this leaf that you can see is yellowish in color, it is experiencing senescence. So the video is going to show you how easily the old leaf comes off from the main stem, and in contrast, the young leaves, that's the green leaves, are very difficult to break off from the stem. Observe. Came off easily, and now when we try to pull on the green leaf, it doesn't come off. Okay, now this happened because we look at the abscission layer. For the green leaf, the abscission layer is very strong. So it does not separate, the petiole cannot separate from the main stem. But in the aged leaf, this abscission layer would have almost disappeared. So that is why it is very easy for the petiole to fall off. I'll share with you another video to explain another factor. In this video, you will notice that when we pull out a young leaf, the part where the abscission layer has been separated will still release some liquids, some plant sap. But when we pull onto a leaf that is experiencing senescence, the abscission layer would be dry. Observe. You see now, the stem here already starts to release some form of latex. okay, showing that that part of the leaf is still very alive. Now look at this leaf that is senescent. Okay, there is no liquid whatsoever coming out. Okay, this is because the abscission layer also has the vascular tissues. So in a young leaf, these vascular tissues are still actively transporting. So when we forcefully break the petiole, the sap that is being transported will still be present and will start to move out of the abscission layer. But if we are talking about a plant or leaf that has senescent, the abscission layer would have started to become loose and the vascular tissue here would have shrink. So when the vascular tissues here shrink, the sap or liquid in the vascular tissues will remain inside the main stem. The petiole will have no more vascular tissue liquid or sap in it. So that is why it will appear dry. Now that you understand abscission, let me move on to dormancy. So the role of abscisic acid in inhibiting cell division, inhibiting cell growth, I am not going to elaborate because I've already discussed cell division and cell growth when introducing auxin and cytokinin. Similarly, the seed dormancy, which is opposite to seed germination, I'm also not going to explain here because previously when introducing gibberellin, I've told you I will explain about seed germination in the following chapter, right? So we go on to the part about closure of stomata. Now you know plants naturally open their stomata for transpiration to occur, and this enables transport of water and cooling down of the plant. But if the sunlight is too extreme, then the plant will begin to lose too much water, causing it to wilt. This you have been already introduced to is called water stress. So during water stress, the stomata has to close. So this is made possible by the hormone abscisic acid. And you've learned about this previously in chapter 6 when we studied gaseous exchange. You can view this process in my video on the opening and closing of stomata. So I will not repeat that in this video. So with that, I've concluded our discussion for abscisic acid. Let's move on to the next hormone. Start off with a question. Can you tell me how you ripen a mango quickly at home? If not you, how do your parents do it? Okay, I suspect these may be the suggested answers. 1. You may put the unripe mango in the container where you store rice at home. 2. You may just wrap it up in a paper bag and store it. Or if you have no paper bag, you will use the newspaper. Now, have you ever tried packing the mango together with a ripe tomato or placing the mango together with ripe bananas? All of the methods above will actually ripen the mango. Now, the reason is because in the case of rice, ripe tomato, and ripe banana, they release a gas known as ethane. And this gas turns out to be a hormone, which helps to ripen the fruit. Now, use of paper bag and newspaper is not that they release ethane. It is just that packing them in material like paper bags and newspaper help to increase the concentration of ethane because you are preventing the ethane from diffusing out easily. So the ethane will be always surrounding the fruit, causing the fruit to ripen. So that is what we will be discussing now. This is the molecular structure of ethane. Ethane is the only hormone that is in the form of a gas. Ethane is also called ethylene. Referring to our summary chart from our video on plant hormones, Introduction to Plant Hormones, you find that ethane has three functions here. That is in the process of flowering, in the process of fruit development, as well as abscission. Let me now take you through the details of each stage, starting with flowering. Now, ethane is able to break bud dormancy and in this way stimulate flowering. Okay, first let's see the word dormancy. Whenever we come to the word dormancy, it is going to be connected to abscisic acid because abscisic acid promotes dormancy in every aspect of the plant. Okay, it prevents the plant from growing, prevents the fruit from developing, prevents the flower from blooming. So ethane, although it is also an aging hormone, but you find that it can be antagonistic to its partner hormone. So when abscisic acid concentrations are high, the flower will remain as a bud. Bud means it's like closed. But once the ethane concentration increases, the bud will break dormancy. That means it will like come alive and start to bloom. Okay, so this is an example where the hormones are antagonistic. Then we move on to fruit development. Now previously we've learned that auxin, cytokinin and gibberellin are also involved in fruit development, but in a process known as parthenocarpy, development of seedless fruits. Okay, but in the case of ethane, it doesn't make the fruit. The fruit is made by the first three hormones, and we find ethane will ripen the fruit. So like the mango earlier, if it is green, then it is because the ethane concentration is low. But once it starts to change color, then the ethane concentration is increasing. This function only ethane can carry out. The third is of course abscission. We've learned about it. So you find ethane promotes abscission. Defoliation. Defoliation means the falling of leaves. Foliage. Foliage is leaf. So defoliation is the falling of leaf. And of course the aging process, that is senescence. So here ethane functions the same way as abscisic acid. But of course it is antagonistic with all the young hormones, that is auxin, cytokinin and gibberellin. The function of ethane, let me explain a little more about the functions that are specific to ethane, that is flowering and fruit ripening. Now the flowering process will be covered in another subtopic, so I'll focus on the fruit ripening. Now when a fruit ripens, there are three things you will notice. Firstly, a ripe fruit has a softer texture than an unripe fruit. This is possible because the hormone ethane will stimulate enzymes that will help break down the cellulose in the cell wall of the fruit. So that is why the fruit becomes easier to bite when ripe. Second change is the color. You will notice unripe fruits are usually green and ripe fruits become yellow, red or even purplish in color. The reason is because ethane will stimulate enzymes that will break down the green chlorophyll pigment in the fruits. And the third thing, the reason why we love fruits so much is that when you bite into a ripe fruit, it is sweet. Now, ethane will again activate enzymes that can break down storage nutrients such as starch. This starch is tasteless. That's why unripe fruits can seem tasteless or sour. Now, the enzymes will break down the starch and whatever other storage nutrients into monosaccharides. And monosaccharides provide the sweetness to the fruit. That I conclude the story of the two aging enzymes that are involved in the development of a plant. So I'll see you in my next video. Let's go have a break. Bye-bye.